Okay, so we left off talking about sodium-potassium pump, and we went through this diagram and explained how this worked. And again, I reminded you and urged you that you need to know the steps of this very, very well. And remember that mnemonic, water follows salt. So let's move on in lecture. And this is another depiction kind of what we talked about. So in a steady state or a homeostatic state, we talked about homeostasis a little bit. We'll talk more about it. The rate of salt transport is equal to the rate of salt import, and water would move out and in with it, and so these would be kind of equal. And like I said, we would be in a isotonic state. Our cells would be in an isotonic state, and we would be in what we call homeostasis, okay? Homeostasis. That's sort of the body's normal. And then they show in this second portion, when the rate of sodium leaving exceeds inward diffusion, then if sodium is going out faster, then water is also going out faster, because water follows salt. Water follows salt. And then in this third one here, if salt is going in at a faster rate than it should be, and it's going in faster than it's leaving, then water is also going in at a faster rate. Water is, because water follows salt, and the cell swells. And so they don't do it, it's not like huge and giant, but you can see that this cell is shriveled a little smaller than this, what they would consider normal state cell, and then this one here is expanded a little more than what they would consider of this normal when compared to this normal cell over here on the left. So that's just the idea that we talked about, water following salt. And then let's move on. Okay, so we did talk about isotonic, hypertonic, and hypotonic. So we talked about it in the example. If you remember, I gave you an example about the dialysis tubing inside of a beaker of water, right? The dialysis tubing, we'll draw that here, inside of this beaker of water. And we said that if the concentration of solution inside was equal to the concentration of solution outside, we had what was called an isotonic situation, right? An isotonic situation. And then we said that if the concentration inside was greater than the concentration outside, that this solution would be hypertonic in relation to the solution in the beaker. And then of course the last one, the third one, if the concentration inside were lower than the concentration outside, then we would say that this solution inside of the pouch here would be hypotonic in relation to the solution on the outside. So that's a review of how we talked about it when we were talking about the beaker of water and the semi-permeable dialysis tubing. And you'll do that experiment in lab if you have lab this semester. Now, as I said, we can apply that to the cell. So you can have an isotonic extracellular fluid which maintains cell volume. But I want to stress here, if it's isotonic, what it's doing is it's maintaining normal cell volume, okay? Normal cell volume. So that's when the tonicity of the cell, relative concentrations of the solutes in the two fluids, both inside and outside of the cell, are relatively equal, okay? Normal volume. Then when we have pathologies or changes or variations in tonicity for either unhealthy or healthy reasons, we can see either hypertonic or hypotonic extracellular fluid. So when the salt concentration, we'll just stick with the salt example, in the extracellular fluid, meaning outside the cell, increases, then we have a variation in tonicity. Water follows, and the cell does what? It shrinks. Now, if it shrinks too much, it can't function. We're squishing the organelles and we're impeding proper movement and function of the cell, and you can actually shrink a cell enough that it will die. You can shrink a cell enough that it will die, okay? That would be a major pathology. All right, and then on the flip side, we can have a lower than normal salt concentration in the extracellular fluid, lower than normal, which means that the inside salt concentration inside the cell is higher than the salt concentration outside the cell. And so water will follow. Water will go inside the cell in order to try to even things out. And then you're going to see swelling. And what will happen in extreme states is that that cell may swell and it would actually burst, or we sometimes call that lyse. So if you see that, the cell would lyse and burst, and then of course the cell would die. So either variation in the extreme context can cause the cell to die or misfunction, but in the extreme most case, to die. All right, so then this is just another image depiction. Here they show a red blood cell. We know it's a red blood cell because it's got this nice round shape with this biconcave center on the both sides, and in the center we see this sort of central pallor. So we'll talk about red blood cells later when we get into the blood section, and you'll see them in lab. But this is a red blood cell, and this is a red blood cell in isotonic environment, and you could see it's exactly normal. It's exactly how it should look. But then if you drop it into a hypertonic solution, which means that there's increased number of ions outside the cell here, water's gonna follow, and the cell is gonna shrivel and die. And then, of course, if you drop it into a hypotonic solution, then there's more ions inside. Let's change color because that's kind of hard to see. Let's change color. There's more ions inside the cell, more solutes. And so water's going to follow, and then this is going to swell. And potentially, if it swells enough, it can burst, okay? So just another example for you. So hopefully you're getting this. Okay, so let's talk then about how we transform matter and energy. So the body, we're going to look at a term here. I'm going to highlight it for you, called metabolism. So this is the idea that we either collect or burn energy, and the net sum of those collections of energy or burnt energy is called metabolism. So it would look like this. Let's say we accumulate 50 ATP and we burn 45 ATP, and it's sort of the sum total. We're left with 5 ATP, and this process, the collecting and the burning, is called metabolism. Now let's say that let's break that down even further. We're just looking at some terms here to get started. When we utilize ATP, it's called anabolism, okay? We are taking small molecules and we're making big molecules. So when would be one instance in when we would do that we've already talked about. So just as a reminder, protein synthesis, whether that protein, wherever it's being used, if we are putting it together, we are taking small units, we called those amino acids. We're linking them together, amino acids. And as soon as we get at least 3, we have a protein. The process of adding and linking all of these together utilizes ATP. It uses the energy of ATP, and so ATP goes down, or we subtract ATP, and that is called anabolism, okay? Utilizing ATP is called anabolism. Now, when we break down large molecules, we are breaking those bonds, and we're providing the body with more phosphates, and those are going into the mitochondria, and they're being used to produce ATP. So we get an increase, a net increase in ATP, and when that happens, it's called catabolism. So the entire process linked together, the creating and expenditure of ATP, is called metabolism. And that's what we refer to when we're exercising and we're dieting. We're talking about increasing our metabolism, the turnover of ATP going and coming. And when we do that, when we're exercising and dieting and we increase our metabolism, then we are breaking down some of these high energy molecules in our body, like fats and triglycerides, and we are dieting and we're being successful. Okay, so that's the idea behind metabolism. All right, now we're going to talk a little bit about enzymes. So all of these processes in our body, of course, if we are taking ADP and we're breaking it apart, or let's see, in this instance, we're putting it together, we are coming up ADP. Let me get my marker back out here. We're taking ADP, we're adding a phosphate on there, which creates a high energy bond, and we're coming up with ATP. And that is a chemical process, right? Because we've got these molecules out here, and maybe this is our adenosine, and then we've got, let's draw this on our other screen. These are chemical equations. Let's get rid of this. So we've got this adenosine molecule out here. We'll just put a big A on that. And then we add to it, we've got on here two phosphates. Actually, they aren't really ions because they are sharing electrons here, but we've got two phosphates. And then out here, we've got this little phosphate ion. See if I can draw that a little clearer. This phosphate ion, which comes in here and has a chemical reaction. And then this adds on here, and by doing so, we change ADP into ATP. And that's a chemical reaction. These are molecules that are reacting with each other. So the idea is that an enzyme, for every chemical reaction in the body, every biochemical reaction, we have an enzyme that is required to work towards that reaction, okay? And they're very specific. If we're making ATP, there's an enzyme for that. Or along the process, let's say that there's, 6 steps to making ATP. There might be 6 enzymes that are involved in facilitating or speeding up that process to get to ATP, but every single one is specific. So if there were one enzyme for this entire reaction, it wouldn't work for a different metabolic reaction. It only works for this one. And if there's one enzyme that converts step 1 to step 2, it's the only one that works for step 1 to step 2. And you can't take it and place it in step 3 to 4 because it only works in step 1 to 2. So enzymes tend to be very, very specific. All right, so we talked about how we break down our energy and our resources. So we're going to talk a little bit more about that, starting with glucose and what glucose does for us. But we're going to pause the video here and then come back with the third section, okay?